Very much for being here. So my talk will be about the system we have developed at the National Center called FACTA Plus. And to give you the background, this is a system that which uses several text mining tools to do text mining semantic search, and also to extract direct and indirect associations. So as we know, several text search engines which are quite important for extracting knowledge from literature. And by that we mean extracting semantic metadata, different types of semantic metadata like name entities, or in this case facts or events, produce different types of systems, of search systems. I mentioned certain systems there that are already existing in the domain. So what actually the focus of this work, we're based on FACTA, and the FACTA is all online. And by the way, I wanted to give you an online demo to show that the system is really real time, but unfortunately there's no internet connection, so it won't be the same, I'm afraid. Maybe we'll 75%. So the work of FACTA Plus is based on a previous system we have done, which actually finds associations between concepts using different co-occurrence statistics. This work is already online, is on our website. Both systems are on our website. And we try to find now the indirect associations, very much very close to what Swanson has suggested in the 90s. These indirect associations and hidden associations are quite important for knowledge discovery and hypothesis generation. So FACTA Plus focuses on 6 biomedical concepts: genes, diseases, drugs, symptoms, enzymes, diseases, and chemical compounds. But it allows you to have flexible queries. It's a real-time response system, which I could have demonstrated if I would have had internet connection. And the functionalities, the novelty of FACTA Plus is that we introduce search using biomolecular events, and I'll explain what is an event, the indirect associations, and also a more interesting visualization. And this is the screenshot of our system, FACTA Plus. So I will start by saying what is, first of all, why do we extract events, and why do we need to extract events, and what is an event, first of all. This is a bit more like a text mining speak. But here's a sentence from the last shared task we have developed. We hypothesized that the phosphorylation of TRF2 inhibits its binding to the CD40 cytoplasmic domain. So what we care here to extract are events like negative regulation, binding, and phosphorylation. And of course to do this automatically. So in this case, when you're doing your query, your search system does not only extract bioentities like genes or enzymes or drugs, but also you can specify that you're interested in phosphorylation of a specific gene or protein, or a drug or an enzyme. In addition, you can do other things, which we don't do right now, like understand, for instance, if an event is speculative or not. So the biomolecular event types we have annotated, actually we have produced, we allow the user to use are the following 9: gene expression, transcription, protein catabolism, localization, binding, phosphorylation, regulation, positive and negative regulation. And those event types, they have been specified from a shared task we have developed in 2009, the BioNLP shared task. It's very close to, for the community, it's like a kind of sister to BioCreative, but with somehow more perhaps linguistic focus. So those are, in case you don't know, you can find on our different websites. The 2009, one of the tasks of the 2009 BioNLP shared task is to extract certain types of events. And we use those events to be able to allow the system to query entities and event types. So if you want to see a bit more about the complexity of the task, this previous sentence here, you have a negative regulation, which is a kind of top event, but includes and has certain trigger words like inhibits, and includes other events like phosphorylation and binding. And as you can see here, the triggers are like different verbs, inhibits or to bind, or nominalizations like phosphorylation. So those are exactly the triggers we're using to be able to extract the events. For the last BioNLP shared task, we have also just finished another one in 2011, which again introduced extraction of event types from full papers. So how do we recognize events? We use triggers, and those are some examples, although resting circuit cells express. So you can see here the triggers for gene expression, for positive regulation, for phosphorylation, and negative regulation. So in text, in FACTA Plus, when you're extracting the events, you see also the triggers appearing in text. So you're extracting concepts, but also types of phosphorylation and negative regulation. So in this case, the event triggers, as I said, indicate the existence of events, and they're used as event indices. So how can we do that? And is it easy? One can say, okay, then let's produce a dictionary, and based on a dictionary, we can actually use verbs and nominalizations, nouns, as triggers. But the problem with dictionary-based approaches are that very often they produce false positives. Of course, as you will see in the paper, in the literature, other people have suggested different ways to use dictionaries and improve the extraction of triggers for events. Why it is ambiguous? If you look at the verb express, you can have various meanings. It can be used as a positive regulation or a gene expression and so on. So in our case, we opted to use a machine learning approach, and we used the training data, the shared task BioNLP training data from Genia, who's from the University of Tokyo, for training, and also used conditional random fields, and in this case, a joint approach, joint learning of event triggers and protein names. The hypothesis of that is, if you have the triggers, as I said, phosphorylate, verbs, and nominalizations, and if you have, for instance, proteins occurring together, and if you actually learn together, you have better performance. So in this case, you will see the CRF, we introduced 3 tags, the B for protein, and an I for protein, and yes, positive regulation. But in addition, we used fillers. And we show by this approach, by using the filler, as I'll explain later on here, represents actually regions between the protein names and the trigger words, which belong to the same event. So it actually helps, it's like a glue, it helps you establish a link that the trigger word is connected to a protein name. So by using the fillers, the CRF model allows to propagate information from any trigger words to any adjacent, in this case protein names, but one can use any type of name entity. So what is the performance of that for extracting, detecting events? You can, by evaluating, if you use only the triggers, you can see the performance. It's just, for instance, the trigger entities, verbs and nominalizations. Joint is the verbs, nominalizations, and the protein names. And this is with the fillers, and you have quite a good improvement. So the reason we are doing that is to boost the performance of event extraction and to show that contextual information, in this case from protein names, has been useful in detecting trigger words for events. And these are the results. I'll show, and on the paper you will see with other ones with joint or not joint. You see here the results by using the joint and filler information for the types of biomolecular entities that we have identified. As you can see, the performance differs depending on the type of event. In certain cases, like protein catabolism, you have a good performance, and in some cases, like for instance regulation, is quite depressed. The reason this is depressed is because certain events are very complex. They include other cascaded events, and very often you need much more rich information. In this case, we don't do that, so we're relying on a very kind of simple approach to extract those events. But if you want to improve, you have to invest in a bit more extracting complex events. So this is what I said before, and about the evaluation and the quality of indexing on FACTA Plus was to actually manually checking 10 latest articles by biologists, and 86 out of 90 abstracts were relevant. So it's okay for the precision. For the recall, still a bit unclear how we could do that. Now, I'm sorry we don't have Wi-Fi. I cannot actually show you the demo live because it would have been better. But a screenshot of that, if you are, for instance, here, you look at FACTA, the gene expression, you're extracting expressions, triggers like expression, overexpression, and so on. So basically it shows you the events from the whole of Medline. So those are the types of events. So that's the first part of the improvement of FACTA Plus. So you don't only do your search based on name entities like genes or proteins or drugs, but you can actually demonstrate, you can make a query using also events. The second part we worked on is how to discover indirect associations, and that's very much according to the philosophy and the ideas of Swanson in the 90s. Now the reason, if you look at the first system, the FACTA, the associations were found by direct co-occurrence. So this is what we call old knowledge. You find these associations in text. But the interesting thing is to see not the direct, the indirect. So associations which are not necessarily co-occurring together. And this is again the case that if X is associated with Y and Y with Z, then the potential association between X and Z is considered as a good hypothesis, except if you have already an existing association between X and Z. So here you have a query. Your query could be not only, again, the name entities like a gene, but also the event. So you can extract, as we say in the paper, E-cadherin and gene or positive regulation. So you can actually specify, you can do your associations between the pivot concepts and the target. So you're extracting associations via something else. And the way that the indirect associations are actually, I'll show it here, is it really works on the amount of new information, the surprising information that you have to the user. So here you have the P1 and P2 is how probable the indirect association is. In this case, how surprising the indirect association. So if in the demo I would have shown that if you have a very high number in the surprising, in the indirect association, then it's actually new information, the novelty. So you have basically X and Y between P1 and P2, and the element of surprise is here. So low, P3 is old knowledge, and very high is probable new knowledge. So thankfully I've prepared some screenshots. So here is an example of indirect associations. So here I put the E-cadherin, and as you can see, E-cadherin is occurring. This is the query, and you can specify also an event. And it shows, and it has a very high element of surprise into the first column here, 13. So it is indirectly associated with diseases like Parkinson's and so on, via those specific concepts. On the top, do we have any? I cannot show. So here, for instance, you can specify that it's not only genes on the pivot and the target concepts, but you can specify the different 6 biomolecular concepts. So here you can actually specify, in this case I put gene and protein and disease, but you can put, for instance, symptoms or enzymes and so on. But here is the interesting thing. Here is the element, as I said, is associated with different diseases via these specific genes. And in this case, you have the indirectly associated concepts are ranked by the expected amount of new information. And here is another example of a prepared with an event. So you have a different re-ranking because now you incorporate event information, and that's here with negative regulation, the same, and you have a different type of ranking. Okay. because it takes into account the event type. You're searching with a different information. So those are the concept dictionaries we have used, in case you're interested. And let's go to the third part. So initially, as you saw before, we had columns, and those were a bit not very interesting and intuitive for the user. So the third part we have done is to visualize those indirect and indirect associations using tree mapping. And this is based on Snyderman's ideas. So basically, I don't know if it doesn't work, all right? Well, it's a shame. So for instance, for Facta Plus, you have, I'll show you here, okay? So this, for instance, this box, this rectangle is quite big because this is quite important. So you can see the importance about the size of the boxes. A big box means important, not so big means not so important. And then this is again with Ikaterin and negative regulation, but it's actually you can try it after when we have internet, because the systems are on our website. So obviously Ikaterin appears often with cancers, and then here you can actually you can actually choose one of the proteins, which is Cas4, and you can pin the relation with the types of associations. So clearly here is working with, with Parkinson's disease, Alzheimer's diseases, and so on. And the, how do you call it? The different types of relations links here depend on how important is the score. So if you have a thin line, that's sort of the length, the depth of the line depends on the types of associations, if it's important or not. So in this case, you can see immediately that this specific Ikaterin is indirectly associated with nervous system disorders, for instance, Alzheimer's disease, Parkinson's disease, and epilepsy. I cannot do that, but there are two types. The first one, you can use the Facta just to see with columns the direct and indirect associations. And if you're in the direct associations, you can also go and view the documents as well, directly the snippets of information from Medline. And in this case, you have the visualization, what I showed before. Okay? So I'll conclude now. So basically, the Facta Plus is based on an existing system we had on finding direct associations from the whole of Medline, but in this case based on 6 different biomedical concepts, and actually it's a different way of ranking the information. In this case we have added also biomolecular events, so in theory more events can be added as long as we have some training annotated data. So the novelties is actually how we can improve detecting event triggers using CRFs by using a joint learning method of event triggers and protein names and adding the fillers, which improve their performance. The second part was discovering the indirect association concepts, and in this case we used a kind of simple, because there are other people doing that, an efficient mining of two-step associations. And we ranked the concept space of the element of surprise for the new information. And last, the visualization was actually to find a more intuitive way to show to the users the association strengths by tree mapping. And acknowledgement, this is funded by BBSRC. Thank you.